Yes, sir. Welcome to the good morning, one and all. Welcome to the third day of the faculty development program on enhancing faculty expertise in pharmaceutical sciences and clinical education. I now request Dr. M. Sudhakar Sir, Principal and Professor at Mallareddy College of Pharmacy, to deliver the welcome address for this esteemed occasion.
Respected Nagarajan sir.
Sir, good morning. How are you, sir?
Sir, very happy to see you after a long time.
Yes, sir. Yes, sir. Sorry for the delay. Yeah, due to rain, little technical glitches. That's the reason.
And after a number of, I think after 2 decades,
Yes, sir. Yes, sir. Yes, sir.
Very happy to see your lesson again.
Sure, sir.
So it will be helpful to all the fraternity all over.
I expect the same, sir.
Great lecture. Please, sir.
Okay, sir. Shall I proceed?
Madam, you introduce, sir. Madam. 1 minute.
Yes, sir. No problem. Because here I'm not able to see the camera. I can able to see only the presentation. Hello.
I request Manoranjani to introduce JSK Nagarajan sir to all the participants. Please start.
At this moment, I am greatly honored to introduce our keynote speaker, Dr. J.S.K. Nagarajan sir. Dr. Nagarajan sir is a seasoned pharmaceutical scientist with a distinguished career spanning decades. His academic journey, marked by impressive credentials from renowned institutions such as JSS Academy of Higher Education and Research and Annamala University, has provided him with a robust foundation in pharmaceutical sciences and marketing. Dr. Nagarajan sir's expertise extends far beyond academia, evidenced by his extensive experience in conducting bioequivalence studies and his pivotal role in clinical development initiatives. His leadership at JSS College of Pharmacy, UT, including his positions as the head of the Drugs Testing Laboratory and Deputy Director of the Center for Advanced Drug Research and Testing, underscores his invaluable contributions to the field. Throughout his illustrious career, Dr. Nagarajan sir has actively engaged in numerous academic and professional endeavors, including the organization of seminars, symposiums, and workshops. His dedication to mentorship is exemplified by his guidance of countless postgraduate and undergraduate students. Additionally, his role in disseminating knowledge through media appearances and research publications has further established his reputation as a highly respected expert in the pharmaceutical sector. I now request Nagarajan sir to deliver the lecture.
Thank you, madam. First of all, I'd like to thank, yes, sir. Thank you, sir. First of all, I'd like to thank Dr. Sudhakar and coordinators for inviting me to give this presentation. And also, I'd like to thank the management for the opportunity given. So here, I'm going to discuss about liquid chromatography technique for analyzing drugs and pharmaceuticals. Also going to discuss about the calibration and method validation. See here, why I have mentioned the strategies, because many of us make small errors when we go for analyzing drugs and pharmaceuticals. So what I'm going to do is, I'm going to start with the basic presentation and almost I'll follow the other strategies, validation and calibration. See, whenever if you look at the picture, you can say why and how do we separate. See, like this picture, our drugs and pharmaceuticals are complex in nature, right? See, if it is a single drug, it is not an issue, but nowadays, a lot of fixed dose combinations are also available in the market. So, how do we go for quantifying the sample? So, chromatography is the versatility technique to separate the components. So before going for chromatography technique, there are a lot of separation techniques are available. Right, it starts from filtration. This is also a separation technique. Distillation is also a separation technique. Evaporation, sedimentation, centrifugation, decantation, sealing, and magnetism. So these are all almost separation technique. How far the chromatography is differ from these techniques? So here also, see, if you look at these pictures, we are utilizing the physical principle to separate the component, whether it is a solid liquid or liquid. So different principle we adopt. Almost same kind of principles. Okay, we are also adopting in the case of chromatography technique. So if you look at the mix of components, right, you can separate the two different ways. One is by chemical separation, other one is by physical separation. If the compound is homogeneous in nature, okay, so like for example, vinegar, nothing but water and acetic acid, it is very difficult to separate in the sense of normally we cannot go for it. We have to go for treating with the other way, we have to go for it. And we cannot able to see here because it is almost gelled with water. The acetic acid that has become a homogeneous solution. But if it is heterogeneous water and oil, if you look at the example now, okay, we can able to separate by using a simple extraction glasswares. Right, but similarly, if you go for physical separation, it is very difficult because almost it is in the form of solid or it is a liquid. Okay, we have to utilize some of the techniques. For that only we are using chromatography. And chromatography, as we know that, okay, it's a separation of mixture of compounds into the individual components. Right. So whenever we talk about chromatography, right, normally we do say it is developed, you can say discovered or developed the chromatography techniques, right? But it is not so because he has developed only thin layer chromatography. But if you look at the ancient times onwards, we have handled, we are utilizing thin layer chromatography techniques. Right? So on what basis I'm just quoting this example is nothing but see, there is a proverb in Tamil, in Hindi, even Telugu also we say, right? For example, here you can see a bird, right? This bird, it's like a swan, nothing but, right? So if you mix the milk with the water, right, you get water and milk. If you give it to this swan, okay, it drinks only milk and not water. It separates water like that. It's a myth is there, but it is not so. This bird is not having that kind of activity. But here we talk about annam in Tamil or in other languages we call it annam or anna, something like we say. But if we look at the rice is separating. So, for example, the same thing, water and milk. If you mix it, okay, use this rice as adsorbent. Okay, the rice will take milk and not water. So the milk is separated. Sorry, the milk is separated by the pure milk is separated by the rice. Okay, so that is what this rice, this quote has been used since from long. Right, so that is what I say. It is not discovered from the ancient times. We talk about chromatography. daily basis, we are seeing many chromatography techniques like this. So, as I said, the principle is mixture of compounds into the components. By after that, you can go for analyzing it, identifying it, purifying it, and finally you can go for quantification. And based on three different phases, solid, gas, and liquid, we can classify the chromatographic techniques. That is principle. Okay, solid, liquid, you can go for liquid gas, and solid gas. Here I have highlighted with the red mark. Okay, that I'll tell you later why. So if you look at the solid liquid chromatography, solid stationary phase and liquid mobile phase that we use it, and for examples are column chromatography, TLC, HPLC, exchange ion exchange chromatography, vapor adsorption chromatography, we can say example. And in the case of liquid chromatography, both are liquid in nature, and here a simple example is paper chromatography, or is called as paper partition chromatography. So in the case of paper partition chromatography, right, paper has got a small pores on that. Okay, so in that pore, the water will be almost formed on a thin layer. Okay, so that acts as a stationary phase and mobile phase that normally we are using the solvents and water. And in the case of liquid gas chromatography, normally gas chromatography, liquid stationary phase and gas mobile phase that we are using it. And in the case of solid gas chromatography, is it possible? Because gas will not penetrate and move along with the solid. So what is the example we can give? That is the example here, right? So what is that example? Nothing but tooth powder. Okay, tooth powder is a solid form, right? And when you go for brushing it, so at the time of brushing, you are cleaning the buccal area. You can say solid liquid chromatography. Also, a buccal area has got some bad odor will be there. Okay, so the adsorbent here, what is the adsorbent? Normally we can say calcium carbonate. Calcium carbonate is a adsorbent that absorbs the bad gases. So once again, since from long we are utilizing the adsorbent to separate. So that is what solid gas chromatography is also possible. That is one example. And another example also that I can quote, nothing but charcoal tablet. Okay. So now in charcoal tablet, when we are using, if you feel any gastritis or gases forms in the stomach, no, that if you want to remove it, okay, the physicians are prescribing charcoal tablet. So charcoal tablet is in the form of solid, and the gas, whatever is there in the stomach, can be removed. Okay, so solid gas is also, we have example, and this example once again, since from long ancient time that we are using it. Okay, there are five different principles that we are adopting to separate the compounds. Okay, one is adsorption technique, second one is partition, third one is ion exchange, and fourth one is molecular exclusion and affinity chromatography. So adsorption chromatography, as we know, the basic TLC, common chromatography, HPLC comes under this category, and partition, you can say paper chromatography as I mentioned before, and ion exchange here to separate the ions, we go for it, okay, by using the resin. And in the case of molecular exclusion, we call it as a sieving chromatography or filtration type of chromatography. You can say here the different size of the molecules can be separated, and larger molecule will be excluded and smaller molecule will be traveling along with the gel. So larger molecule will be easily separated when compared to the smaller one. And the last one is affinity. Here, the organisms, okay, biotech products can be separated. That is the example, right? So if you have chromatography, five different chromatography that normally we are using, right? In the sense, first example is first example is paper chromatography. As we know that this is the very inferior one, okay? And second one, we go for TLC. And third one, you can say column chromatography. And the fourth one is HPLC. And and the last one is GC. So 5 different technology that we go for adopting it. Okay, and directly now I'm going for HPLC technique. And as I told you, HPLC comes under absorption technique. And there are different modes that can be used, reverse phase mode and normal phase mode. And mixture of analytes can be separated. It depends upon the attraction towards the stationary phase. If it is highly attracted, travels slow, and not at all attracted, moves faster. And no two analytes have a similar affinity. In that case, there will not be any separation. So even if you look at the instrumentation part, okay, there will be a solvent reservoirs. Then we have to go for mixing. After mixing, it goes to the pump, and after that goes to the injector. Okay, and then goes to the column, and finally go to the detector. So these are the different components that we have already studied. Okay, right? And types of HPLC technique when you are thinking about it, as I mentioned, okay, based on modes. Normal phase mode, you can prefer it, and next one is reverse phase mode, you can prefer it. The other one is elution technique. Here, isoparetic way also separate. You can separate the components. There is a mix of components into individual, and gradient separation also you can go for it. And principle, as I mentioned, 5 different. Apart from that, chiral compounds also can be separated. Chiral in the sense, there will be, for example, optical isomers. If the compounds are optical isomers, minimum two isomers will be the D and L. Okay, if you want to separate D and L, you can go separate it by using highly specific isomeric column that can be used. And then scale of operation, you can go for it, analytical HPLC and preparative HPLC. And here, finally, that is based on type of analysis, qualitative and quantitative also can be performed. Most of the time, now what I'm going to do is I'm going to discuss only right now is for analytical HPLC. So when you look at the HPLC, as I told you, it is between solid and liquid. So when you consider about the solid, okay, adsorbents are required, right? You might have studied in thin layer chromatography. Okay, the adsorbents are classified into 3, namely weak adsorbent, medium adsorbent, strong adsorbent. So whenever we want to go for HPLC, we use strong adsorbent like silica gel. Even alumina is also available, but alumina has got a little issue. Alumina can be classified into alumina basic, alumina acidic, alumina neutral is there. But if we look at silica gel, it is highly neutral. It is nothing but silicon OH group will be there. Slightly, you can say, 2 weakly acidic compound. So it is not going to play a major role. So you can consider one way silica gel is a neutral compound, right? So column, this is the picture of the column that I am representing here. So it depends upon our analysis, okay, whether it is for a preparative or analytical purpose or microbore. Okay, accordingly, the column dia and the flow rate also will have to vary. Okay, the length of the column also varies, depends upon that. Okay, right? So normally once again, what I'm going to say is we are going to go for analytical column. So column once again, okay, we will be having guard column, analytical column, preparative column. So preparative column is meant for herbal compounds, okay, for the extraction, separation of compounds in the bulk, and analytical column, we go for quantification, a guard column to separate the unwanted material we use it. Okay, so if you look at the analytical column, as I mentioned here, okay, I'm going to discuss about only analytical column. The length normally, okay, 10 to 30 centimeter, it is meant for normal HPLC. If it is UPLC, you can go for 5 centimeter column also. And the column will be normally straight. Okay, since there is no curve or something like. Because why I'm saying is, in the case GC, we use circular columns like. Okay, also the column, there are many numbers are there. You can connect 2 columns also. Okay, continuously, that is series like. And if you look at the dia, 4 to 10 mm is required. And in the case of normal HPLC, the particle size will be 5 micron. And for the case of UPLC, we have to use less than 5 micron, that is 3 micron or 1 micron also you can go for it for the better resolution. And as you mentioned, 25 centimeter column. So finally, the requirement for normal HPLC technique, okay, basically what we do is we go for 25 centimeter length, 4.6 mm diameter, and 5 micron particle size. So if you look at the blades, okay, 40,000 to 60,000 plates per meter we go for it. So it depends upon the purpose. We can choose either microboard, analytical, or preparative. And the next part is liquid. Okay, what is liquid? It is a mobile phase. To separate, okay, we are supposed to use solvent or buffers, right? So the choice of the solvent, the mobile phase is very important in the case of HPLC because it is going to elute, it is going to separate the components. So it is all based upon the polarity of the solvent as well as polarity of the compound and polarity of the sample, stationary phase. And the solvents, okay, both we can use inorganic solvent as well as organic solvent. Inorganic solvent, example for inorganic solvent is water, and organic solvent as a listed alcohol and pentane. Even you can go for paraffins also, like, okay, in that sense what I'm coming to say is pentane, sorry, not even pentane, you can go for liquid paraffins, not liquid, sorry, you can go for sorry, I'm not getting an example, okay, ethers also you can go for it, pet ethers, okay, right, so, and once again, the solvents are classified into polar solvent. Simple example, okay, whichever the compound has got OH and H and SH is called as polar solvent. And in the case of non-polar, we have decane, hexane, pentane, cyclohexane. There is no functional group, only parent components are there. Right? So this solvent, selection of this solvent is important by in the sense it has to pass through the column because we are going to separate a compound into components, individual components. Right, so since it is packed, we need to apply a pressure between 1000 to 3000 psi. Okay, and also the particle size is smallest one. And technique-wise, you can go for isocratic separation gradient. Isocratic means throughout the experiment, we are going to use only one solvent. In the sense, pre-mix the solvent, you can go for it. Okay, two mobile phases, you can mix the required proportion and you can go for it. But gradient here, two different solvent reservoirs will be there, so you can choose the ratio using the system. You can go for program, you can go for it. So here I have given a list of solvents and solvent strength as well as polarity index I have given. Right? So while using the solvent, degassing is important because it has got some gases inside. So there are possibility of vacuum filtration, you can do it, helium budging, you can do it, ultrasonation also you can go for it. So to remove the degassing, better you can go for ultrasonation. And if you look at the developmental techniques, so what we have discussed is, first we have discussed about stationary phase, mobile phase, okay. Next is developmental technique, right? So if you look at paper chromatography, there are 5 different chromatography developmental techniques are available, okay. And so before that, right, we have TLC, we have HPLC, we have column, and we have position. Okay, right. So for the example, paper chromatography, we have 5 different development techniques. It is ascending, okay, it's due to capillary force, and descending like gravitational, right? And ascending, then descending was the third one. Force, right? After running, you have to change the absorbent 90 degree, then you can proceed for it, and as well as radial chromatography. And almost if you look at TLC, okay, ascending you can use, radial you can use, as well as 2D you can go for Column only one possibility, it is nothing but descending chromatography. There is no possibility of ascending. Okay, because you have to collect the elements, okay, at the bottom. How about gas chromatography? So gas chromatography, once again, being gas, okay, it moves from bottom to top, right? And what about HPLC? HPLC is whether ascending or ascending, descending, or 2D. Which mode we can go for it? HPLC, which method? That's a question. There are two methods normally we can go for it. One is ascending. Okay, second one is 2D. Right? Why I'm saying ascending? Okay, that's the reason we are using pump. If you look at gas, okay, if you look at the gas, it is ascending only, but easily the gas move from bottom to top. You will not do anything. But liquids will not move from bottom to top. We have to go for pumping it. Though adsorbents are there, though capillary action we talk about here, paper and TLC, the large amount of adsorbents are there, as well as it is very tiny. So here we are going for only ascending technique. Apart from that, you can connect up two different columns continuously. Okay, right? Like here, what we do is we never change. Instead of changing the point, here we change the mobile phase ratio. Okay, mobile phase and mobile phase ratio. And in the case of HPLC 2D, what we do is we connect two different columns. Maybe you can have the same mobile phases. Okay, suddenly all of a sudden you cannot change the mobile phase. So that's the reason we are changing the column, not the mobile phase here. So this is normal representation, how it works, okay? The HPLC, right? So it's an example I have given, okay? But the picture shows here differently, right? Okay, this is for the representation only, how this has been separated, right? And once separation is over, what is final? Detection. So detection once again, choosing the detector is the most important part. How to detect? It's all depend upon, okay, the organic compounds or in whatever way. So one is based on bulk property, and second one is based on solute property. So in the case of bulk property, we can go for refractive index. Normally for bulk property, we go for refractive index instead of dielectric constant on density. And in the case of solute property, okay, there are many possibilities. It is nothing but you can go for derivatization. Okay, after derivatization also you can go for it, or without derivatization also you can go for it. Right? So as per IP, if you look at, right, most of the time we use U. Okay, it is connected to Q. Otherwise, what you can do is hypnotic technique, you can go for PDA and mass spectrophotometry. Okay, these are the detectors can be used. And if you want, if the compound is very small quantity, if we want to go, there will no other possibility in the sense in that case. Right. So this is a different type of detector. And what is the sensitivity? What is the amount of compound that can be limit of detection possible, and linearity that I have given here. Okay, I listed out. So as I told you, UV is the widely used one, particularly for drugs and pharmaceuticals, because most of our drugs are having benzene in the structure. Okay, so being benzene in the structure, straight away you will not have second thought. Okay, without having second thought, you can go for selecting UV. Okay, right? So when you prefer UV, specifically we go for only one particular wavelength. But if you prefer PDA, you can select many number of absorbents, many number of wavelengths that you can select, 5 wavelengths, 6 wavelengths also. So you can study about all five wavelength, which peaks are good, where the resolution is good. So automatically you can find out instead of going for changing the wavelength each and every time. So UV is the only disadvantage is that when compared to PDA. Okay, so that is what PDA is useful. So whenever you go for UV and PDA, right? Selection of the solvent is more important because solvent sometimes it absorbs the light. Okay, so the cutoff wavelength first always solvent should be less than 200 or something like that. Okay, so less than 200 if you go for it. We have acetonitrile, methanol, and water we have. So these solvents normally we prefer to go for UV or PDA. And after that, okay, we get a peak, and this is nothing but representation of a chromatogram. So based upon this chromatogram, okay, you can go for quantification or qualitative analysis. So qualitative analysis based on RT, that is retention time. Okay, same, almost similar retention time that we are getting or not. Okay, in the case of quantification based upon the area, peak area, you can go for comparison between the standard and sample. So that is what normally we use the following parameters. In the case of HPLC, that is retention time, retention volume, separation factor, resolution, height theoretical field plate, efficiency, and symmetry factor. So here, the compound, if it is a single compound, not an issue. When we get multiple components, okay, very difficult. See, for example, here little separations not happening properly, right? Here the resolution is very good. So such a way that you have to select the mobile phase and everything, right? And whenever we go for analysis, there are different peaks we come across: broad peak, ghost peak, negative peak, and peak doubling. Okay, these peaks, okay, normally you get it. So it is by experience, you can sort it out. Okay, broad peak is what is the problem. Concentration is high, or the flow rate is low, or what. Likewise, you can find out ghost peaks, similarly negative peaks, absorbance related one. Okay, so based on that, we can find it out. And these are the applications. You can use it for chemical, pharmaceutical, clinical, consumer, bioscience, and environmental. Okay, so this is nothing but a short information about HPLC, right? And what is next? Strategy. How we are going to develop the method. See, so far, whatever the thing I said, solid phase is required, mobile phase is required, the detector is required. Okay, right? So these are the three basic things. So based on that, we have to prepare the strategy. Right. So before go for it, so here as I mentioned, drugs and pharmaceuticals. See, when you look at pharmacology, chemistry, pharmacology, okay, whenever you study, they start with the classification of the drugs based on the plants in the case of pharmacy, based on this organs or something like you can say system, in the case of pharmacology. In the case of chemistry, based on ring, you can classify. But how do you classify drugs in the case of analysis? So far, no one is talking about this. That is the problem in the case of separation of compounds. Because first we have to study about what type of drug is it and pharmaceuticals, right? So if you look at Indian pharmacopoeia, we have many number of drugs are available. It is validated. The methods are being also validated. It is enlisted in pharmacopoeia as per alphabetical order. Okay, so when you are looking at IPs again, the drugs, inorganic drugs, organic drugs, and even plants also will be represented. Okay, right. So when I'm talking about drugs and pharmaceuticals, here I will concentrate on organic compounds, not inorganic. Okay, so inorganic we have, instead of going for chromatography, easily you can detect a different metals are available. For example, copper, zinc, magnesium, some mercury. If anything is there, even it is in the form of mixture, you can quantify, not only identify, quantify by using atomic consumption spectroscopy, or otherwise calcium, sodium, potassium mixture is there, then you can use flame photometry also. But for organic, okay, you have to classify the drugs. So how will you classify the drugs for organic? Right, 1 minute. Okay, so the organic compounds are classified as weak acid, weak bases, two weakly acidic compound, two weakly basic compounds. Right? So once again, and few neutral compounds are also available. In IP, what is available? Most of the drugs are two weakly basic. Right? It is called heterocyclic compounds. So it is nitrogen-based compounds. Nitrogen-based compounds are two weakly basic. The exception is whenever the compound undergoes ketonal-tautomerism, these compounds are called as two weakly acidic compounds. Simple example, you can go phenobarbital, that is barbiturates, phenol. These are the examples. Right? So why am I talking about? Not only the classification and also the drugs are polar in nature. Right? So most of the drugs will have OH group or NH group or SH group. Okay, you can find NH and OH lot. So once again, drugs and pharmaceuticals, organic drugs and pharmaceuticals as per the AP are highly polar in nature. So that you have to keep it in mind before we proceed for the separation technique. So we have to select the stationary phase, is the drugs are polar or non-polar. And based upon that, you have to select the mobile phase. And what type of solvent that can be used. You have to select the flow rate, temperature, pH, and developmental technique. Which developmental technique you need to go for it? We are supposed to go and detect that. Okay, so these are the major things you have to keep it in mind before we go for it. And finally, derivative decision technique. So how will you classify? That's what I said. Okay, weak acid, weak bases, and compounds are also there, right? But most of our compounds are two weakly acidic compounds and two weakly basic compounds. So whenever two weakly acidic and two weakly basics, okay, you can say the compound is polar in nature. So the compound is polar, that is, drug is polar. Okay, it should not connect, it should not attracted by the adsorbent. Okay, so which mode we have to go for it? Reverse phase mode we have to go for it. Okay, because the drug is polar. If the mobile, if the stationary phase is polar, then the delay in processing, in the sense separation will delay. Okay, so that is what instead of selenol OH group. Okay, what we do is we remove that OH or H. Okay, nothing but alcohol. Our alcohol is converted into silane. Okay, we call it as methylated silane. So here I have given an example C4, C8, and C18. C4 means nothing but 4 carbon is connected with SiO. That is SiOH is converted into C4, SiO. And next, instead of C4, C8 is possibility, and C18 is also you can go for it. Apart from that, you can go for cyano and amino column. Amino column, whenever hydroxyl groups are more, that time we go for amino column. Otherwise, okay, the first preference goes to almost, you can say, C18. Okay, it is highly non-polar, right? So reverse phase mode. So stationary phase is reverse phase. And mobile phase, as I told you, inorganic as well as organic solvents are there, right? So water, methanol, ethanol, ACN, likewise n number of solvents are there, so you can go for it, mobile phase. So mobile phase will be almost polar in Okay, that is what it is meaning. Right. So similarly, depends upon the combo that we can go for selecting the mobile phase, stationary phase, mode, as well as the type of compounds. It's all based on that. See, for example, if you look at size exclusion, okay, here we use a gel, right? The mode is nothing but size exclusion, stationary phase is silica, polystyrene, silica with a small orifice will be there, so that a smaller compound will travel. Okay, so this is about the drugs. Right. Next is stationary phase. Okay, so most of, as I told you, most of the drugs are polar in nature. So as I mentioned the previous one, C18 is the better option. You can refer IP almost whichever the method they have shown, they have enlisted, okay, they have documented. All are based on C18. Otherwise, they call it as ODS, octadecylsilane column. Okay, that is the column only we have used. So once you have selected the stationary phase, okay, being the drug is polar, we have to select C18, right? Then we have to go for selecting the mobile phase. So mobile phase on what basis, right? So whenever we talk about mobile phase, compulsorily two mobile phases are required. One is organic, another one is in aqueous. Okay, aqueous means doesn't mean water, right? You can go for buffer. So it is nothing but a combination of organic solvent as well as buffer. So in the case of organic solvent, right, the best option is ACN and methanol. Why I am talking about here ACN and methanol? We prefer to use detector which one UV or PVA. So in that case, these solvents will not interfere and easily will gel with water even. That is buffers also. So that is the reason that we have to select a proper organic solvent. Which one we are going for is nothing but methanol or ACN, or you can go for combination also. The next one is buffer. Okay. Buffer, once again, inorganic buffers are available as well as the organic buffers are also available. Okay. So in the case of inorganic buffers, we can go for phosphate, and in the case of organic, okay, it is almost weak when compared to phosphate, citrate, acetate, formate. So selection of buffers, okay, is most important. Okay, most of the time, if the simple way you're going for separation, okay, you can go for phosphate buffer. So within 3 minutes, 4 minutes, the drug will elute. Okay, and sometimes may not be suitable for bioanalytical method validation. Okay, and because it is like I can say interaction between the two acidic compound as well as inorganic. Okay, it is a strong acidic kind of thing you can say when compared to citrate. So our drug is weekly and go for weekly buffers. Okay, weekly basic salts or weekly acidic salts you can prefer it. Right? So in that case, acetate, formate, and citrate you can prefer it. Okay. So when you prefer citrate or acetate or formate, the same method can be adopted for bioanalytical even. Okay, if you use for phosphate buffer, we cannot go for it. That is one. And the phosphate buffer, suppose you are connecting with the mass spectroscopy. Okay, phosphate is not ideal. Better you can go for citrate, acetate, and formate because phosphate now easily they reacts and thereby a lot of other issues starts. Okay, and buffer selected. And what is the buffer? Ionic strength. Okay, so we have to select 10 millimole maximum we can go for it. You can be able to separate very nicely. Right? So once it is over, selection of mobile phase. Temperature also we need to select. That is column temperature here we am talking about. The column is placed in the compartment. Okay, it will be controlled by can program to the temperature even whatever the temperature 140, 50 or something like. Better try to go for ambient condition. Okay, so ambient condition is always advisable by the compound sometimes degrades. Okay, so whenever you want to go for separation, you can avoid using going for higher temperatures. Okay, so once temperature is also over flow rate. So flow rate maximum you can prefer 1 ml. You can go for even up to 10 ml, but there will be back pressure will be possible. So that is the reason you can go for maximum 1 ml. And apart from that, we have to select because we are using buffer as one of the mobile phase. So we have to select the pH of the buffer. It's also important. Okay, so how to select the pH? Based upon the pK of the drug, plus or minus pH can be used. For example, the pK is 9. Okay, plus or minus 2 is 5. Okay, 11. So pH 5 and better. Like that, we have to go for selecting the pH. Okay, and this is about the flow rate. The flow rate is over, temperature is over, pH is also over. So next is developmental technique. Okay, so in the case of already have shown, in the case of paper chromatography, 5 different development techniques are available. The case of TLC 3, and in the case of column and GC only one ascending one is descending, another one is ascending respectively. And in the case of HPLC, we have only 2 options. Okay, one is ascending, another one is 2D. Okay, that is what here, ascending and 2D. Nowadays, even people are going for scouting. Scouting in the sense you can use 5 different columns also. Okay, the same, so the mixture of compounds will be passed through all 5 columns. In the sense, it is not like a fractional distillation, you can say, simple example, scouting. Okay, for, the same kind of mixture will go to individually, and finally you can be able to select it. Right? So you have selected station phase, you have selected the mobile phase, you have selected the pH, temperature, flow rate, and developmental technique. That is ascending or 2D. The final one is not the final before that. Okay, data is important. So always advisable. If you're going for basic method development, U is better. Okay, or even better, you can go for PDA. Nothing but photodiode or data. But if you want to go for, okay, bioanalytical, mass is better, right? And whenever you are developing a method for regulatory purpose, okay, you can go for mass, right? So if you prefer these 2, possibility of cheating, you can show the same configure for some other compound also, because it's very difficult to find out, but not in the case of mass. The compound molecular differs. So if you have mass spectroscopy, it is better, right? And finally, derivatization, whether we need to go for derivatization or not. So according to me, what I will say is better you can avoid derivatization, because derivatization, when you talk about pre-derivatization and post-derivatization, that is converting a compound into another compound by using a reagent. So whenever we go for derivatization, 5 to 6 parameters are there that affects our analysis. For example, phenol, okay, reacts with the ferric chloride, gives violet color, right? So violet color, on what basis I'm getting it? What is the concentration of the reagent? What is the volume of the reagent is required? In which condition, heating or cooling? In which condition, the other way, it is acidic condition or basic condition or neutral condition, right? And you got the color. Derivatization is converting the colorless compound into color. And that is a simple example you can say. So when compound is converted into colorless to color, how long the drug is stable? Okay, so these are all you have to study. Then only you can be able to. So what happens if you are not going for derivatization, your method will be completed in a simplest manner, right? If the compound is complicated, in that case only you can enter derivatization. Right? Normally what we say is fluorescence, you can go for it. In smaller quantity you can able to quantify. Otherwise, you can avoid going for derivatization. Okay. And so once method has been developed, you have selected stationary phase, mobile phase, you have selected the flow rate 1 ml, temperature is ambient, ambient is nothing but up to 21 degrees centigrade. Okay, here why I'm saying ambient, not room temperature. Room temperature differs from place to place. For example, Hyderabad may cross even 45 and Ooty in the summer it will be 25. If we consider Kashmir, okay, 5 degrees centigrade, right? So you cannot say room temperature. Here you have to say only the ambient temperature. Ambient temperature is controlled temperature. The laboratory will be controlled between 19 to 21 by air conditioning. Okay, so pH is maintained, then go for developmental technique. So normally we go for ascending, not 2D, right? And then detector as it will UV, okay, PA and mass. So advantages I have given. Derivatization as I told you, better not to go for it, right? So once the method is developed, we need to validate. Why we have to validate? What is the reason? Okay, see, whatever the method is developed, is there any scientific evidence? Scientific sense is there or not? Okay, and whatever the method that I have developed, okay, the same method can be handled by some other people also. So that is the way we have to see the method, right? So once again, if you refer IP, whatever the method is available, it is validated. Okay, it is validated by the regulatory people. In the sense, not drug inspector, they might have given to one of the other laboratories, central laboratories, they might have given and they might have developed. Okay, the same thing you can use it any part of the world, right? So if you look at the analytical method validation, there are around 10 parameters are here, and these 10 parameters are classified into two Qs I have given. One is qualitative, another one is quantitative parameters, right? So here for analytical, okay, specificity, selectivity, robustness, and ruggedness. Then LOQ, linearity range, accuracy, precision. So in the case of qualitative, okay, we talk about specificity, selectivity, robustness, and ruggedness, and quantification. We go for this LOQ, linearity, range, accuracy, precision. So let us see one by one. Okay. This is the example I have given. Once again, this is from ICH guidelines. Okay. So what is specificity, selectivity? And nothing but simple example I can say. Phenol plus chloride gives violet color. So this violet color is specifically for phenol in presence of chloride. Okay, so that is what specificity is the color, selectivity is the chloride and neutral condition. Okay, so that is the way we go for it, right? That is specificity, selectivity. So here we are not going to measure. We are not going to measure what is the amount of the component is present. And robust and ruggedness. So when we are talking about robust, okay, whatever the selectivity and specificity is selected, okay, there is no changes. The color or whatever will be. The solution, okay, stable for long duration. See, some of the compounds easily oxidizable, like in fidepine, nitroglycerin and all. You have to perform the assay or quantification in a dark room or something like. Okay, other drugs not required. So easily, whichever the compound easily get oxidizes. So you need to use it. You have to perform the method in the dark rooms or something like you say. And even pH little changes when you make it. Okay, like pH, okay, plus or minus 0.5. And mobile phase composition, little variation, 95, 5, for example, you have selected two different mobile phase, two different solvents, okay, 96, 4 or 90, then something like small changes that you can make it. By changing the small changes, okay, it should not affect the specificity and selectivity. Similarly, temperature and also flow rate. So flow rate normally what we fix is 1 ml per minute. You can go for plus or minus 0.1 ml. Okay, this is nothing but 0.9 to 1 you can go for it. So there should not be any much changes. And ruggedness is nothing but different laboratories, different analysts, different instruments, different days. Different laboratory, for example, that's what I said, ambient conditioning. When you are performing ambient conditioning, okay, the method can be adopted, that method can be used in different laboratories and different people also. Because once the procedure is given, the same thing should get it, that is repeatability should be there. And instruments, different make instruments are available, water, smash, okay, even if you change the instrument, you should get the same result. And this, okay, that's what stability I'm talking about here. The next parameter is LOD, LOQ. So LOD, LOQ, almost you can say L is quantitative like. Okay, it is like LOD, LOQ. Why we are talking about LOD, LOQ? Whenever we perform chromatography, okay, and detection is by spectroscopy. We are using spectroscopy. It is connected to the electricity, right? So whenever we connect it to the electricity, there will be a noise like radio. Okay, for example, when you want to listen to radio, we go for 108.1. For example, when we say this is the frequency, so we have to set 108.1 exactly. If you slightly here and there, okay, if you go for it, what will happen? You start getting noise. Okay, similarly here also. We have to identify which is signal and which is noise. For example, much problem may not be there in the case of analytical, but when you are going for bioanalytical, drugs may be present in picogram level in analytical fluids. So almost it is resembling to the noise. So you have to identify. Okay, so on what basis? So the ratio, the noise is 1, okay, the signal must be 3. The ratio is 1 is to 3 or 3 is to 1. SNR, we say SNR, signal to noise ratio, 3 is to 1. Okay, height-wise, we can go for non-instrumental based on visual evaluation, or we can go for the other techniques also by using instrument also, you can go for it. And quantification, from where we can say quantify. Can you able to quantify? Here you cannot be able to quantify. Yes, drug is present or not, you can say. But here you can start quantifying. That is nothing but SNR ratio will be 10 and 10 is to 1. This is nothing but noise, 10 times more that of noise, that is signal is 10 times more than that of noise. So here you can start quantification. And then comes LOD and LOQ. You got in the sense, okay, the next step is linearity and range. Okay, so here we have determined what is the quantity that can be detected, and from where we can go for quantification. So for example, 10 microgram I can able to quantify, right? So linearity above 10 only you have to fix it, below 10 is not possible. So linearity you have to select. Okay, so whenever we talk about linearity, you can go for 5, 7, 9. Okay, odd numbers you can select because to reduce the errors. And minimum, in the case of analytical, you can go for 5, and in the case of bioanalytical, you can go for 9, you can go for it. That is based on Cmax. Okay, and here we can go for percentage 50, 100, 150 or something like that. So the term linearity itself says when you increase the concentration, automatically the absorbance, the pKa will also increases proportionally. That is what here. Okay. So after performing it, we have to analyze statistically. It has to give regression analysis 0.99 is there. Then the method is suitable. Otherwise, the method is not suitable. We have to go for calibration and range. As I told you, there are linearity. 5 different concentrations that we are choosing. One is lower concentration, another one is upper concentration. So any concentration that you can go for it. Microgram, picogram, milligram, also if you want, you can go for it. See, as to what I have seen, selectivity, specificity, selectivity. Second one is robustness, ruggedness. Third one is LOD, and now completed is linearity range. The first two is nothing but specificity, selectivity. Robustness, ruggedness is related to qualitative, and we talk about LOD. From this onwards, it is nothing but quantitative. The last part is accuracy and precision. Okay, right? So here, accuracy, because it is we prepare the solutions, standard solutions. Okay, so whenever we prepare the solutions and we keep it as a stock, we say stock solutions, right? So we have to get repeated rate. Right, accuracy also we should get it. So that's what we select three different concentrations: QC, HQC, and each and every concentration we go for three replication. So similarities, that is almost accuracy, closeness to the value we should get it, like you can say accuracy and precision. So the limit is 99 to 101 in the case of drug substance, and in the case of drug product, it is 98 to 102. So that is what here it said. Okay, repeatability. So once that is over, okay, so your method is developed. Okay, so almost I just discussed around 5 parameters, namely specificity, selectivity, robustness, ruggedness, LOD, LOQ. Okay, and then linearity, range, precision, accuracy. Right? If this is over, your method is fit for the purpose. If anyone almost quantification, accuracy or precision is not matching, you have to find out okay where you have made a mistake. See, normally in academics, most of the time, what we do is we don't calibrate the instrument, and even we don't calibrate the glasswares. Most of the institutions may not have class A apparatus. Okay, so whenever you want to go for regulatory purpose, compulsorily you are supposed to use the glasswares also should be calibrated. Right? Even for example, I have mentioned that we are going to discuss in the later slides. Okay, so glasswares to be calibrated, instrument also to be calibrated. Okay, so now the third part is nothing but calibration of HPLC. Okay, that is nothing but instruments. Why we have to calibrate? See, normally we are using different chemicals, okay, right, like phosphates, many things. So whenever we use chemicals, it is contacting with the instruments. Okay, somewhere spillage and many things happens. So automatically there will be possibility of damage, right? And sometimes in the case of UV, we use the light source, right? See, for example, at home, we are using tubelets. At the end, you can see the blackened particle. It is nothing. Okay, the fluorescent material is getting oxidized. When the time increases, okay, the material get oxidizes. Same thing incandescent bulb. We call it as normal bulb. Okay, it is connected with the filament. Right. So over longer duration, sometimes the filament get disconnected, right, due to longer usage. So that is the reason you need to go for calibration. Not only source, wherever, okay, we are talking about accuracy, precision, and all, we have to think about it. So the first one is gradient check. Second one is, okay, first here this is related to separation, gradient separation. Whenever we talk about gradient separation, we have to think about gradient check. And then we go for injecting the sample, right? So injector is also should have accuracy. Then flow rate, we are using pump. Pump may not be properly working now. Then in the case, we have to check the accuracy. Then column. So here temperature, okay. Thermometer. Whenever you wanted to go for checking the column moment temperature accuracy, whether whatever the temperature that you have used, okay, 40 minutes it is 40. It is okay, only you can go for it, otherwise no. So here the thermometer also should be calibrated. And finally, wavelength. So wavelength is related to detector. So this is detector related, and this is nothing but column oven related, and this is nothing but pump related. This is nothing but injector related, and this is solvent reservoir. Okay. Right, so here we go for linearity checking, both the side. Linearity means you have to go for 5 different concentrations, you have to prepare and find out whether it is passing the limit. Okay. So gradient accuracy. So in the case of gradient accuracy, right? So there will be two channels. Suppose isochaotic, no issue. We are mixing both the solvent as well as buffer, and then we go for pumping it. But here we use two reservoirs, A and B, right? So here, for example, one is methanol you have used, another one is caffeine or something we have used. Okay, the absorbance will vary between these two. Okay, channel 1 and channel 2. So you have to check the absorbance by fixing the particular wavelength. Then injector. Okay, so different concentration we inject, whether in the loop, okay, it is almost 10 microliters or 20 microliters occupying, that we have to study. So that is what injector linearity. So as I told you, 5 different concentrations. Okay, we have to take from 0 to 100 microliter, inject it, and whether we are getting the linearity graph or not. Not only linearity graph, the value correlation coefficient should be 0.999. In that case only it is positive, otherwise it is fails. So in that case you have to check it out, where is the problem. Okay, our concentration, many things you can say. Then flow rate. So flow rate is when we say 1 ml, okay, it has to pass 1 ml from one end to the other end of the column, right? Because that checks the performance of the pump, okay? Because once again, pump easily get damages, right? That's the reason, because of the mobile phases. So how to test? You fix 2 ml per minute, collect 25 ml in a standard flask, and then go for weighing. Okay, 2%. The limit can vary plus or minus 2%. This is about creating injector and flow rate. The next slide what you're going to see is, and this is the last slide. Okay, column oven temperature accuracy. Okay, here you have to select 2 different temperatures. Okay, and study it. Okay, right, and that is it. Calibrate thermometer here, you have to keep it. Okay, what is the reading you are getting it? Right, the reading must be plus or minus 2. These changes can be there, not more than that. Right, and finally, wavelength accuracy is related to the detector. Right, so here also we prepare standard solutions. Go for checking the wavelength. What is the nanometer it is showing? Okay, right? So plus a 2 nanometer that can be available. That is what here. So taking the blank as less than standard. So wavelength is almost we'll be fixing on 255 nanometer. Okay, sometimes 256, sometimes 257 will be there. Okay, we change for even. So that's the reason here. So today I just briefed about the chromatography, different types of chromatography. Okay, and then I've concentrated on HPLC, right? Just highlighted the basic information, and after that, I just selected only HPLC. Okay, right? Because most of the trucks we are preferring HPLC, not even GC, very limited. Whenever you wanted to analyze a solvent, HPLC is not the right technique. So in that case, only you can go for GC. Otherwise, you can be comfortable with HPLC. Right? So I've given the strategy how to select the stationary phase, mobile phase, what is the flow rate we have to fix, temperature is required or not, and pH, what is the pH, what is the detector that you have to allow, fix, what is the developmental technique. So after that, I've discussed about that, briefed about method validation, because method validation is the mandatory one. Okay, because whatever the method you have developed is called as in-house method. It is okay. It is not regulatory based method, in-house method. So whenever you develop method, in-house method, compulsorily you have to go for full validation. Okay, and then I discuss about calibration of HPLC. Right. So based upon the components, starts from reservoir till the detector. So that's all about the presentation. So sorry, detector is also the same thing. Okay, system precision, you can go ahead with that. So almost same thing with the linearity, injector linearity, no? The same thing here, we are going to inject it. Okay, right. So that's it from my side. Over to Sudhakar.
Sir, Nagarajan, sir.
Yes, sir. Echoing, sir. Yes, sir.
We had a beautiful session.
Thank you, sir.
Our JSK professor, Professor K. JSK Nagarajan. Thank you very much, sir.
Thank you,
Any
sir.
questions? Just you text in the chat box.
Okay, sir.
Give the answers.
That we can do it later or now itself, because here I'm not able to see.
Just I'm checking, sir. If any questions are
Yeah.
there,
Okay. Okay.
right.
Because being YouTube, no, people will ask later on also sometimes.
Yes,
Because it anyhow it will be in the streamline, no.
yes, yes. So you are taking a lot of effort to have this session. Yes,
Because weather, sir, due to weather condition,
so it
the
is
disturbance
useful.
always will be there for us. And that too hilly area, so we don't get network connectivity. So once again we are changing our even Wi-Fi systems also.
Good.
Thank you, sir, for being with us
Thank
today
you.
and
Okay,
detailing
ma'am.
about
Thank you.
cryptographic strategies and HPLC.
Okay,
We
ma'am.
are certain that your insights will enrich this program, sir.
Thank you, ma'am.
The second session shall resume at 2 o'clock.
Okay, ma'am.
Thank you, sir.
So shall we leave, sir? Sudhakar, sir.
Yes, sir. Welcome, sir. Welcome. Please.
Yeah. Thank you. Thanks for the opportunity once again to Dr. Sudhakar and coordinators as well as their management. Thank
Yes.
you.
We'll meet personally. Nagaraj, sir. Thank you all. Initially, we had some technical issues and other points. Finally, successfully completed in one session. Thank you all. Thank you very much.